Hello and welcome back, everyone. My name is Sarah Brain, and I'm an assistant professor in the sociology department and the PRC. It is my honor today to introduce Professor Elliot Tucker-Drob as our first PRC brown bag speaker of the semester. And this presentation, I wanted to note, is in collaboration with CAPS, or the Center on Aging and Population Science. So Professor Tucker-Drob is a professor in the psychology department here at UT. He is director of the Lifespan Development Lab and a faculty research associate here at the PRC. And in his work, Professor Tucker-Drob asks how and why people progress along different trajectories of psychological development across their life course. And his research really focuses on how social and educational experiences combine with genetic variation to impact cognitive development and mental health over time. His work has been funded by the NIH and has received national and international acclaim, most recently having been honored the Max Planck Humboldt Medal. So I could go on, but I think that you would rather hear from the scholar himself than from me. So with that, please join me in welcoming Professor Tucker-Drob.
All right, well, thanks for that nice introduction. Let me go ahead and share my screen. Okay, so it's great to be here. I think it's been a while since I spoke to the PRC, and it's my first CAPS talk. So I'm going to be talking about kind of a host of related topics related to how I conceptualize cognitive aging and dementia. And let me just kind of get started with my take-home message. And I want to highlight a problem and I want to propose a solution. So the problem is that current practices for diagnosing dementia produce distorted epidemiological inferences. And I'm going to illustrate that quite a bit over the course of this talk. And the solution is that we need as a field to move more towards the analysis of rates of longitudinal change using cognitive measures that are sensitive to the entire distribution of functioning. And I'll talk about what I mean by sensitive to the entire distribution of functioning as I proceed through this talk. So what I'll be doing is I'll be showing you, I really like to think about these topics visually. And so kind of at a conceptual level, what I'll do is I will illustrate ideas using simulated data, but I'll also intersperse those kind of conceptual ideas with hard empirical evidence. And so you'll see in either red or blue, simulated or empirical, usually in kind of the lower corner, right corner of the slide, whether or not what I'm showing you is simulated or empirical. And you know, I hopefully will be clear about that as I speak as well. If you're interested in kind of reading about these ideas, I would direct you to one of these two recent papers. I kind of talk about these ideas generally in my 2019 annual review paper, and then I consider the issue of education and cognitive functioning across the lifespan, more specifically in this group authored paper published in Psychological Science in the Public Interest. So let's give some background. So when I talk about cognitive aging, what I'm referring to is progressive declines across multiple cognitive functions, such as memory, reasoning, or processing speed, that are normative in adulthood, yet heterogeneous across individuals. So cognitive aging happens to everyone, but it doesn't happen equally to everyone. There are many different partially understood etiologies for cognitive aging. That's kind of a major ongoing area of inquiry, is what causes cognitive aging. And so I can't give you the mechanistic explanation in this definition. And in fact, I think it's best to define the outcome of interest irrespective of the hypothesized causes, because we'll be refining our causal understanding over time. So let me give an example of kind of, these are just two items from that are examples of two different sorts of tests that I might use to assess cognitive functioning. in a study of cognitive aging. So on the example on the left, you could see it asks you to complete the pattern with the missing letter. And if you want to give that a shot yourself, you can try it in your head. I'll tell you the answer in a moment. On the right, it asks you to take this kind of representation of a cutout piece of paper, fold it up in your mind, and match it to the three-dimensional representation and find which of the four options is the one that matches. So you can see these are pretty difficult, and these are just examples of two items, and the items vary in difficulty within a test. But I chose two kind of difficult ones to make a point, which is that when we talk about cognitive aging, we're not just talking about whether or not someone can do a low-level task. That's something that's often tested when we are interested in kind of everyday functioning or whether or not someone is close to dementia. But we're talking about a full range that also includes quite sophisticated modes of thought. On the left, the answer is D. And if you wanted to know why that's the case, what you would have had to figure out is you convert the letters to numbers in terms of their place in the alphabet, and then you'd add the first two to make the third. And on the right, I believe, I'll do it on the spot. I believe the answer is A. And there's some ones that look almost like they could be right, and you can really get convinced that they're right, but there's only one correct answer. And yeah, I think it's A. Okay. So those are just two examples. There are many different sorts of tests that are used, assessing many different types of cognitive function, but I just wanted to give you a flavor. So here what you can see is an empirical plot. So this comes from a nationally representative sample of children and adults from across the United States. I think this sample was collected in, I believe it was the late 1990s. It's a cross-sectional sample. And of course, we could probably definitely spend a whole hour speaking about age period and cohort effects. But what I want to highlight here is that you can see that this outcome, which is referred to as fluid reasoning, so it represents a lot of the sorts of types of reasoning that would be required to successfully complete those two example items that I showed. It begins declining progressively on average. So this is on average for the population in early adulthood. Now, we could talk quite a bit about what do the longitudinal data look like. There are problems with both of them. The longitudinal data, the biggest problem that I think is underappreciated is what we refer to as practice effects, which is the fact that if you've seen a test before, even if it was a decade before, you tend to do better on it. Even if you can't remember the test, there's a certain familiarity that makes you do better. And there's various designs to kind of get at that. My favorite is kind of the waitlist control approach, where you only give half the people the test the first time and everyone the test the second time. And you look at whether or not previous experience with a test makes a difference. So, generally speaking, there's really solid evidence that progressive cognitive declines occur beginning in early adulthood. Now, that's not to say that every ability declines. So there's a class of abilities that are sometimes referred to as crystallized abilities. These are things like vocabulary knowledge, knowledge about the world, things that you learn fairly explicitly from culture and from school. And those actually remain fairly stable until about the age of retirement, about the 60s or 70s. What's kind of interesting about this is we often think of these crystallized gains in early and middle adulthood as kind of defying this idea of cognitive aging. And it turns out in a paper that I think should probably be out in the next week or two, what we find is that the people who are declining the most in their fluid abilities are actually making the least gains in their verbal knowledge and their crystallized abilities. So even though on average in the population, people are getting better in terms of their stores of vocabulary knowledge in middle adulthood, the ones who are increasing the least are those ones that are declining the most in things like fluid reasoning. Okay, so that's cognitive aging. How about dementia? So in the DSM, it's kind of been rebranded as major neurocognitive disorders of aging. the kind of, if I had to give the elevator definition of dementia without all of the bells and whistles, it would be substantial decline in cognitive performance that interferes with daily living. So there's two core aspects here. One is that you have to be declining relative to a previous peak in your cognitive performance, and those declines need to interfere with daily functioning. They have to interfere with your everyday life. The dementias, especially kind of within the medical establishment, are often categorized or classified according to their purported etiologies. I find this a bit strange in the sense that we haven't fully discovered the mechanistic basis for these dementias, but we've already started to define the dementias in terms of their hypothesized mechanisms. But the most common one that's been studied and is receiving quite a bit of attention for good reason is Alzheimer's disease. There's other types of dementia, such as frontotemporal dementia, vascular dementia, and then quite a number of cases of people who present with dementia actually present with what's referred to as mixed etiology, meaning that there are biological hallmarks of multiple forms of dementia pathology, such as Alzheimer's markers and frontotemporal atrophy, for example. So how is dementia diagnosed? So in spite of all of this emphasis on the biological etiology, it turns out that in most clinical settings and in most population-based data sets that you'll actually be analyzing to understand the epidemiology of dementia, we're not relying on biological measurements. It used to be said that for Alzheimer's disease, for instance, you could only even measure it post-mortem after someone has had their has died and had their brain examined. Now there are in vivo ways to measure Alzheimer's pathology. So we can give people radioactive tracers that tag amyloid, for instance, which is a hallmark of Alzheimer's disease, and we can look at the amount of amyloid in the brain using a PET scanner. That said, even though that's possible, by and large, the clinical assessment and the assessment for population-based data sets is based on behavior. So commonly there are kind of two kind of classes of clinical assessments. One is dementia screening instruments. So the most popular one is the mini mental state exam. So here's an example of an item from the mini mental. It says, I'm going to spell a word forwards and I want you to spell it backwards. The word is world, W-O-R-L-D, spell world backwards. So you can see it actually requires some of the same sort of mental manipulation that you might be required to do to perform some of the more complex tests that I showed examples of before, but it's an easier sort of item. And in fact, that's one of the harder ones on the mini mental. The other is what's referred to, type of assessment is what's referred to as a semi-structured interview. And so in that case, a clinician, so it might be a neurologist, for example, would interview a patient and perhaps someone who knows them well, like their spouse or their child, and ask them, have you had problems remembering things lately? And they would take a full history, they would ask lots of questions, and based on the information that they qualitatively gather, they would make an appraisal as to the level of impairment across a number of dimensions. And you can see in this one for the clinical dementia rating scale, the CDR, you can see that it's memory that's being assessed. So dementia is very much assessed in most clinical and research settings in a similar way to the way that cognitive aging is assessed, but with instruments that I would kind of say are a little bit more qualitative. And if not qualitative, they're certainly quantitative, but only measuring the kind of lower range of functioning. And I think that's an important thing to keep in mind. What do the data look like with respect to dementia prevalence rates? It turns out that at about age 65, the dementia prevalence rate is less than 1%. It's quite rare. by the time people are in their 90s, it's a very common diagnosis. People are in Western industrialized countries are estimated to about a third of people over age 90 are estimated to have dementia, and that's dementia. There's also question as to whether or not even more people have the pathology that causes dementia but aren't being diagnosed with it, and those estimates can be as high as 70% in the 9th and 10th decades of life. So these data come from a study in the UK, but you see them in almost all kind of Western industrialized data sets. Okay, so what is the predominant guiding model for dementia research? I would refer to the model as a cascade model, and I would refer to it as a medical model. Medical in the sense that there's this strong emphasis not on a continuum of functioning, but on whether or not functioning is pathological or not. And cascade in the sense that the thought is that a series of biological and subsequently behavioral events happens in sequence that unfold over the course of years, if not decades. So in this very famous figure that you can see on the left, which is sometimes it's by a researcher named Clifford Jack from the Mayo Clinic. So they're called Jack curves. What you can see is that there is this kind of sigmoid or logistic function for various types of markers of dementia. So all the way on the left is that red marker, which is amyloid. It's a biomarker for Alzheimer's disease. And the thought is that early in the disease process, amyloid starts to elevate, but the downstream markers, including memory and clinical function, remain completely unaffected. It's only until people progress into this mild cognitive impairment range do things like memory begin to be affected and things like clinical function begin to be affected. So there's this strong emphasis on the idea that the pathology changes first, behavior and everyday functioning is unaffected and only changes at a much closer time point to the diagnosis itself. And this way of thinking is very much baked in, not just to basic research, but to the way that we regulate treatments and drugs for dementia. So the FDA is the organization that regulates whether or not a drug can be approved for a disease or disorder like Alzheimer's disease. And what you can see in their guidance document, which is the document that drives almost all planning by pharmaceutical companies, for instance, when they're designing their clinical trials and testing their treatments, it talks about there being 4 stages of Alzheimer's disease. And the first stage is one in which there's pathology, but no cognitive decline or clinical impairment. It's stage 2, there are some cognitive deficits, but they're only evident on sensitive neuropsychological measures, and they're thought not to have functional consequences. And then only in stages 3 and 4 are there the sort of prioritized effects on cognition and everyday functioning. And the FDA really urges drug developers to design trials that can that can detect differences at that stage 3 level, which is, I mean, that is a non-trivial issue. Because if we conceptualize the disease progression process as occurring over the course of decades, and we're only designing trials that will be approved by the FDA if they have an effect at that late stage, that means that there could be plenty of drugs or treatments that could have been effective at an earlier stage and stopped the snowball before it got big, and as it rolled down the mountain, to give my PhD advisor's metaphor. But none of those drugs are being tested because the FDA is not interested in them. So this is it's a core and fundamental issue is the way the FDA conceptualizes stages of dementia. But this is just one model. the way I think about this model is as a latent model. And I think this is an important point. So as I mentioned, this FDA guidance and the Jack curves, they're premised on the idea that biological pathology can be detected when there are no clinical or cognitive symptoms. And so the idea here is that cognitive function and clinical or everyday function remains flat during the early phases of disease progression and completely undetected behaviorally. But during that time, the pathology accumulates. So I kind of think of this as about, we have a scale, and as we add beads, that can be thought of as the pathology to the scale doesn't change. It only changes once enough beads are added. But during this entire period of time, pathology is accumulating, and the scale is showing no change. And that's the metaphor for the way this medical model works, that there's this biological change, it's latent and unobserved, and it cannot be detected without imaging or biomarker analysis. Okay. We can think then of people who differ in their onset of Alzheimer's disease using this sort of stylized, simulated figure. And so the idea here is that everyone has a latent phase, but they differ in how long that latent phase is, perhaps because biological pathology is accumulating at different rates for them. They begin to decline from a stable level, and perhaps early on those declines go undetected, but after a few years, they reach this lower threshold of functioning in which they start to perform very poorly on the dementia screeners, and the clinical structured interviews start to detect that people are actually having problems with memory and everyday functioning. So that's the thought. And if we kind of think of this clinical threshold as when people get their diagnosis, this sort of figure on the left can recreate the sort of dementia prevalence figure that I showed before empirically, and now I'm showing it in the simulated data. So if we think what's going on is what's happening on the left, we can kind of recreate what we empirically get, which is that the prevalence rate increases with age. Okay, here's the alternative, and this is one that I've been thinking about for, gosh, about 15 years now. And I've been pretty surprised at how little attention it gets, not because people are ignoring my own research, but because it seems to me as an obvious alternative that other people should be thinking about. And I really don't see it very often, which is that pathology begins to accumulate, perhaps early in adulthood. And as pathology accumulates, cognitive function is affected in lockstep. So as people increase in the amount of pathology they have, and that's in this figure kind of color-coded by more dark shading, their cognitive function declines. And so the more pathology someone has, the more their cognitive function declines. In my mind, that makes a lot of sense, especially when you think about the brain. If the brain starts to shrink, I would expect virtually instantaneously with that brain atrophy that people will be performing worse on cognitive tests. It's not that I expect that the brain shrinks, but people go unaffected for 5 or 10 years, and then eventually their cognition starts to be affected. That doesn't seem plausible to me. Okay, so how might we kind of visualize this? Again, this is simulated data. This idea that there is not a latent phase, but there's a continuous distribution of pathology that's associated with a continuous distribution of cognitive change. What I've done here is I've just simulated a distribution, a normal distribution in this case, of change from baseline. And people who were in the top, I think I made this the top 70%. So people who declined either only mildly or not much at all, I treated as the normal group, and the people who are in the bottom 30%, I treated as the severe group. And I just decided to dichotomize the change at that cut point just for the sake of visual illustration. And so those people who have severe decline, who have more rapid change from baseline, are depicted in red. And you can see they reach this clinical threshold at an earlier age. And if that threshold is the threshold beyond which they get a label of dementia, then we can plot that dementia prevalence rate according to age. And again, we see that same pattern that we see in the empirical data, which is that the dementia prevalence rate increases with age. Okay, and these are stylized to kind of highlight the points that I'm trying to make. I'm going to keep elaborating on them. Okay, so we can think of both of these models, the medical model and the kind of continuous model of cognitive aging and dementia, as what are sometimes referred to as liability threshold models. There's a continuous distribution of liability or risk towards dementia, and past a threshold, people are getting a diagnosis. The distinction is that the liability in the medical model that's represented on the left is latent, meaning that people's behavior and their everyday functioning is unaffected before they meet the threshold. They're accumulating those red beads, but the scale hasn't changed. Whereas the continuous model on the right treats liability as something that is observable. It's something that can be measured in cognitive performance. And so people who are declining, even if they haven't reached that threshold, it's not just that they've accumulated pathology, but they've also declined in their memory. Their memory may still be, relatively speaking, good. It may not be a clinical concern, but it will still be impaired, even if they haven't reached the clinical threshold. So that's the core distinction. Okay, so there's an important measurement issue here. And the measurement issue relates back to that distinction that I made between the kind of difficult cognitive measures that have both difficult and easy items on them, and the dementia screening instruments, which really are constitute very easy items. And the idea here is that, so the mini-mental, that's the most popular dementia screening instrument. The mini-mental is scored out of 30. And if you give the mini-mental to virtually anyone who isn't suffering from mild cognitive impairment or dementia, they'll score a 30 out of 30. Sometimes someone will score a 29 out of 30 just because they make a silly error. But for the most part, people are scoring at ceiling. And you can imagine that had there been more difficult items, those people who are scoring on the real mini-mental a 30 out of 30 might be differentiated from one another. There might be some people who, if the mini-mental went up to 50, might get a 47. They're still way above 30. Other people who might get a 35, also still way above 30, but those are very big differences. And so if we had, instead of a dementia screening instrument, if we had a sensitive cognitive measure, we might expect to see something like the simulated data on the left. And in this data set, what I did is I simulated a situation in which people started diverging in their biomarkers at age 50. And in lockstep with their divergence in biomarkers, they started to diverge in their cognitive function. In the panel on the right, I actually used the exact same data, but anyone who scored, in this case, I have the scale, kind of not on a 30-point scale, but on a 100+ point scale. Anyone who scored 90 or above, instead of giving them the score that they actually earned, I just gave them a 90. So I imposed a ceiling effect on the data. And then what we can see is what appears to be a lagged association, a cascade sort of effect, where the biomarker is showing divergence at age 50, but the cognitive function isn't showing divergence between groups until ages 60 or later. So we get this illusion that there's a lagged effect, that the biomarker change precedes the cognitive change, simply because we're using a dementia screening instrument in which we don't have sensitivity to the higher range of functioning. So this is a core issue, and it's easily solved by using tests that are sensitive to the full distribution. If I had to come up with kind of a silly analogy for it, it would be giving anyone who's above 5'5" on a stadiometer a height of 5'5" instead of giving them their true height. I just don't see the point in it. Okay, so this has been a lot of conceptual talk. Let's show some real data. So in this study, which was published just last year, what my colleagues and I did is we looked at whether or not changes in brain structure were longitudinally coupled with changes in cognitive function. And what's particularly interesting about this data set is it's a data set in which over 700 people have been scanned in an MRI. And so they've had their brain structure measured, not once, but on 3 separate occasions, separated in time by a little bit over 3 years each. They've been measured on their cognitive function using sensitive cognitive measures at all 3 waves. And none of them at baseline have dementia, and an exceedingly small number of them go on to get dementia over this longitudinal interval. We can exclude the few that do get dementia. This may relate to the sample being somewhat positively selected because they were screened prior to enrollment in the study. But what makes that kind of a very interesting opportunity is it allows us to look at how brain change relates to cognitive change during this so-called latent phase, or this silent period of cognitive aging, in which what we're expecting to see is the biology to change, but not the cognition to change, if we believe in the medical model. So here's the study design. They're being measured on 68 different regions of interest within the human cerebral cortex. They're being measured on 3 different cognitive domains: visuospatial ability, processing speed, and memory. And the first thing that we did is we conducted a factor analysis of the rates of change. And what I mean by that is that people differ from each other in their brain structure at any point in time, but we're not interested in that. We want to know about how people differ from each other in their rates of shrinkage of the brain over time, so change from baseline. And when we do that, we basically find 3 fundamental dimensions of cortical change. We find a very strong global factor, which just represents the extent to which the cortex is shrinking or atrophying kind of uniformly across all regions. And then we find these 2 additional dimensions, one that represents kind of frontal shrinkage and the other that represents parietal and occipital shrinkage. And so I'm going to focus on this global factor because it's the most salient. And here's the core finding. It's a pretty simple vector of 3 correlations, but it's the correlation between rates of change in brain and rates of change in these 3 abilities: visuospatial ability, processing speed, and memory. And so these are positive. And so what that means is that the people who are declining, whose volume is shrinking the most in their cortex, are also the ones who are going down the most in their scores on the visual spatial tests, on their processing speed, and in their memory. And those whose brains are not shrinking very much are the ones who are maintaining their cognitive function. And so you might look at these numbers, their correlations between 0.3 and about 0.4, and you might say, well, those aren't particularly large. But the thing I want to emphasize here is this is a correlation, or these are 3 correlations between biology and behavior. And I would be very hard pressed to find reproducible correlations between biology and behavior that were bigger than this. This is pretty much the highest you can get when it comes to taking biology and linking it with something that's quite distal, which is cognitive function. All right, the next empirical example I want to give relates to, it's a similar methodology, but the variables are somewhat different. So instead of looking at how the brain changes in relation to how cognition changes, what I'm interested in is how cognitive function changes in relation to everyday functioning. So you might recall that a core feature of this medical model and the FDA guidelines is that people can decline in their cognitive function when they're kind kind of in the healthy range of functioning. And that can be picked up by sensitive neuropsychological tests, but it won't have an effect on their everyday function. And so what I did here is I used, it's an interesting data set. It was a data set that came from a randomized clinical trial that was attempting to train various cognitive functions in order to prevent people from losing their ability to perform tasks of everyday living. So they put a lot of effort into measuring those everyday functions. Because I'm not interested in the treatment, I'm only looking at people in the no contact control group. So this is essentially an observational longitudinal study that happened to be very preoccupied with measuring everyday functions. And when I say everyday functions, I'm not talking about people's subjective appraisals of their ability to live their lives or take care of themselves, nor am I talking about a clinician's ratings of those based on interviews. What I'm talking about are situations in which each participant is asked to perform the everyday task that they would be expected to do in their lives in front of the researcher, and that is scored. So people are asked to pay bills, so fill out an invoice and write out a check in front of the researcher. They're given a bottle of medicine. They're asked to follow the medication instructions and choose which pills to take at which time. They're asked to make change of $1, and they're asked to look phone numbers up in a phone book. And those are just some examples. And so the question is, do changes in the ability to do those kind of indisputably important tasks of daily living correspond with changes in tests of these so-called sensitive neuropsychological measures? And this is, again, this is not in a sample of people who are converting to dementia. This is in a healthy sample of adults, none of whom have either dementia or mild cognitive impairment. So according to the medical model, these people might show some minor cognitive deficits, but it shouldn't affect their everyday living. But that's not what we find. What we find is that the rates of change over time in everyday problem solving, observed tasks of daily living, and timed instrumental activities of daily living are strongly coupled with the changes in this composite index of general cognitive functioning. So this finding is really flying in the face of the idea that cognitive deficits can occur in disease-free, healthy individuals absent changes in their abilities to perform everyday functions. All right, so my interim summary, and then I'll just go on to a kind of short second part, is that we find strong concurrent longitudinal associations between brain change and cognitive change, and between cognitive change and everyday functioning in healthy older adults, which suggests that silent periods of disease progression may not actually be so silent after all. What I want to touch on now is what clinicians would refer to as premorbal levels of cognitive functioning. And when they say that, what I think is heterogeneity in child cognitive development. So what do I mean by that? Well, where people start off, their decline from peak in adulthood is a function of all of the inputs that have occurred over the first 2 decades of life. There's a tremendous heterogeneity in how children learn and how far they go in school. And by the time children become young adults, they differ in their cognitive performance. And that is an interesting observation in its own right, and it relates strongly to issues of social inequality and education. But for the purposes of this talk, the reason I want to highlight it is because what it means is that when we talk about people declining from a previously higher level of functioning, we have to take into account that people's baselines from which they change differ. And that's a core issue if we're really trying to understand cognitive aging, and we're not trying to, and we don't want to confuse it for what's going on during child development. So imagine these 2 people, one who starts off young adulthood with high cognitive function and another who starts off with low cognitive function. The black line is kind of the average. And what you can see is as we follow them over time, the way that I've simulated these data is that they're aging in parallel. They're not differing from one another at all in how much they're losing their memory, for example. But what you can see is that the person who started off young adulthood at a lower level of cognitive function has reached this dementia threshold at a much earlier age than the individual who started off young adulthood with a higher level of cognitive function. And that's a huge source of bias. What that means is that individuals who, for instance, have had lower access to education during childhood are going to start off adulthood at a lower level of functioning that is closer to the threshold. And so they can decline very little and not actually exhibit signs of pathological aging whatsoever, but still get a dementia diagnosis. And alternatively, you can have people who start off adulthood at a very high level of functioning. And even if they decline quite a bit, they still may not get the dementia diagnosis simply because they're still able to perform above that minimum threshold on these dementia screening instruments. So here's a stylized example of how what this might look like for a whole group of people. The red lines represent people who are declining quickly. Those who in blue represent those who are declining more moderately. And what you can see is that even though some of the red lines are past the dementia threshold, there are other red lines that never pass that dementia threshold. So that's what I would refer to as undetected pathological change. And there's also blue lines that are not actually are representing people that are not actually declining rapidly at all, but they're nevertheless getting a dementia diagnosis. So we have this huge amount of confounding due to what medically oriented people would refer to as premorbid levels of functioning. And this has a major effect in terms of how we interpret figures like this. So on the left comes from one study that I think is illustrative. It's the 10-year incidence rate of dementia for individuals who didn't graduate from high school and those who did. So what you can see is that people who didn't graduate from high school have a much higher rate of dementia over a 10-year period. This is a meta-analysis which basically shows the same finding. It's a little bit of a weird figure because it has exposure to low education level, which is a bit weird in terms of the way I think about education. But on the right, you see lower education and higher dementia risk. So this has naturally led people to assume that education is somehow related to rate of cognitive change. I'm not even going to get into the question about whether or not that relation is causal. I'm just interested in whether or not there's a relation at all. Because what these figures show is not that education is related to rate of cognitive change. All they show is that education is related to the dementia incidence rate. So why might that be? On the left here, now I'm going back to simulated data. What I had just shown were empirical data, is what I think many people intuit when they see the sorts of figures that I showed in the previous slide, which is that people differ in their cognitive function fairly minimally in young adulthood, but that the differences between high and low education groups diverge over time, and the low education group actually declines more rapidly, which leads to a higher rate of dementia over time or as as people get older. And this is what Tim Salthouse has referred to as differential preservation, because people with high and low education differentially preserve their cognitive function. And it explains why we would see different rates of dementia. But an alternative explanation is what Salthouse has referred to as preserved differentiation. He was really trying to mess with us when he gave us these two names. There's differential preservation on the left and preserved differentiation on the right. And what you can see with respect to preserved differentiation is that the difference in cognitive performance between people with high and low education is evident in early adulthood, and that difference is preserved over the life course, such that there are no differences in rate of aging, but there are nevertheless differences in the dementia incidence rate, because the individuals with lower education, low attainment on average, have lower cognitive function and therefore closer to the dementia threshold at an earlier age. So how do we distinguish between these two explanations? Well, the first is we need to ask whether it's even plausible that people begin life with different levels of, begin adulthood with different levels of cognitive functioning. And the answer is yes. So it is plausible. Stuart Ritchie and I actually conducted a meta-analysis in 2018 of natural experiments that didn't just seek to look at whether or not educational attainment was correlated with cognitive function, but we actually found that looked at only at studies that used an identification strategy that allowed them to infer that education caused cognitive function. And we found that generally speaking, for every 1 year of education, cognitive performance as measured on IQ tests increased by about 2 points, which is non-trivial. That means that a college degree is associated with about a half a standard deviation of cognitive function. In this paper that I wrote with a whole bunch of authors and scholars who are smarter and more famous than myself, we assessed the evidence for whether or not the education was actually related to rate of change in cognitive performance in adulthood. And what we concluded is that there's absolutely no evidence that education is associated with different rates of cognitive decline over time. On the left is a meta-analysis. It was one of the key pieces of information in our review. And what you can see from the very simple plot from that meta-analysis of over 100,000 people is that education is related to where people start off. In this case, their cognitive performance at age 60, so kind of in earlier late life, but it is not at all related to the rates of change. So you can see these lines are perfectly parallel. But nevertheless, because of the differences in where individuals start, we should expect to see differences in the dementia incidence and prevalence rates. Okay, so you might ask, haven't there been reports of what Salthouse has referred to as differential preservation with respect to education? Meaning, haven't people found that education is associated with differences in rates of cognitive change? And the answer is yes, but if you look at those reports, nearly all of them are using dementia screening instruments. And I can go on about this for quite a bit, but suffice it to say that if you were to take data that look like that produce the trends on the left, in which the aging related curves are completely parallel, and then you rescore the test as if you're just using a dementia screening instrument and not a sensitive cognitive aging measure, you'll get the pattern on the right. So the pattern on the right is an artifact of using an instrument that is only sensitive to impaired functioning versus normal functioning, but isn't sensitive to differences within the normal range of functioning. All this is to say that that my argument is we need to rethink the conventional view. So rather than thinking about disease progression with respect to Alzheimer's disease and related dementias as a binary outcome that occurs kind of according to a kind of survival type or event occurrence analysis, we need to think about benchmarking person-specific change from baseline using cognitive measures that are sensitive to the full range of cognitive function. So that's what I'm doing now in a currently NIH-funded project with a whole bunch of investigators and participating cohorts. And we're specifically interested in trying to identify genetic variants that are associated with the rate of cognitive change as distinct from simply those that are associated with cognitive function at a given point in time. So this is just a little bit of a plug that if you happen to be someone who has access to a cohort with genetic data and longitudinal cognitive aging data, and we haven't been in touch, we'd love to hear from you, because this entire way of thinking, I think very much applies to all correlates of change, including DNA and other types of biomarkers. So with that, I'd like to acknowledge the generous support of various agencies that have funded this work, and answer any questions that you might have.
Great, thank you so much for this wonderful talk. As we transition to Q&A, I just wanted to remind folks that it is PRC tradition to reserve the first 2 questions for graduate students and then open the floor for broader discussion. So I'm going to moderate the Q&A, so if you could use the hand raise function under reactions at the bottom of your Zoom, that would be great, and I will call on you. So we'll start with Chloe.
Thank you so much for your talk. This was so interesting. I wonder how you might think of other factors of inequality that might cause chronic trauma or chronic stress, and how that might relate to the aging process, specifically with differential preservation. Because in my head, I would presume that, like, not only do you start at a lower level, but we know that higher amounts of allostatic mood can affect the body in like pretty severe ways. And so I wonder how you think about that in context, or like if you think about it similar to education or differently.
That's a great question. The way I think about the problem is very similar to the way I think about education. I'm interested in, one, do we need to worry about the exposure being related to pre-morbid cognitive functioning, meaning is the exposure something that's going to affect child cognitive development, access to education, all of the things having to do with educational attainment? If the answer is yes, then we need to really worry about whether or not, when we find that the exposure is related to either late life cognitive function or dementia prevalence or incidence, whether or not that is an association with an aging process or an association that was evident in the first couple decades of life and has just carried forward. I have no reason to discount the idea that things other than education would lead to differences in rate of cognitive aging. I mean, that's the holy grail. It's just that it requires a higher standard of evidence than I think most people have been subjecting these hypotheses to. And I absolutely agree. Stressors that have neurotoxic effects should absolutely be expected to be related to aging-related cognitive decline. And I would love to see more research that actually looked at rates of cognitive change, as opposed to just, here's one measurement taken at one period of time and how it relates to, the worst case scenario, a retrospective report of trauma. But even in a prospective study, if you just have trauma or stressors taken at one point in time and cognitive function measured at another point in time, but not measured longitudinally, then there's a lot of ambiguity in the interpretation. But I think that those are really valuable research questions, and I think that we're going to find those predictors, but we need to be testing them, in a rigorous way.
Thank you.
I don't see any more grad students, so in the interest of time, Mark, you're up.
That's a great talk. Thank you so much. I think I've been stalking you in your dreams when I look at your graphs. I think we first talked about these graphs in like 2007, stylized graphs at Penn State. It's a long time ago. Anyway, what I was going to ask you is that I would agree with you on this education constant decline over time, but I think that the other issue is empirically the rates of decline in cognitive function can look like they're not parallel that's in part because of exposures that happen in adulthood that are going to affect cognitive function as well. And so as we move into looking at issues in terms of like whether the curves are parallel and how much is carried forward from childhood, we have to also be concerned about the adult exposures that can alter them too, because there's heterogeneity. You did a nice job of showing parallel curves, but the curves can actually cross and go in weird ways just because, people with the same education can end up in different walks of life and have different relationships and those kinds of things. And so it's just something that you made me think about as we went through this. But I wanted to thank you. I do have another question though. In your genetic work, are you actually just hinging it on the ApoE allele, or are you actually going and traipsing off into the GWAS area?
Yeah, okay. So let me just say, Mark, everything you said, I completely agree with. All of the issues having to do with observational data analysis apply to longitudinal observational data analysis. And that means that a childhood exposure is potentially correlated with recurrent adult exposures. And that complicates things even further than the way I illustrated them. And I think that's a really important point to appreciate. So thank you. Yeah, so the motivation behind this genetics, this big genetics project is kind of using ApoE as an example of what we're hoping to kind of find more of. So ApoE is really interesting because in 20-year-olds, ApoE, I mean, there have been a few reports to the contrary, but I think that they're mostly just mining noise. ApoE is not related to cognitive performance. It is not a correlate of child cognitive development. It is only related to cognitive performance as adults get older. So it's a variant that is kind of the perfect example of differential preservation. People start off, people with the E4 allele, which is the version that's risky, they start off young adulthood at no disadvantage relative to people without the E4 allele, but their disadvantage begins to become evident as they progress into adulthood. And of course, there's lots of important research on what might mitigate and modulate those effects, which I am very interested in. What we would like to do is the GWAS that have been conducted on Alzheimer's disease thus far they've detected ApoE and they've detected these other variants, but those other variants that they've detected, it's not clear to me express that pattern in which they're not correlated with early adult function, but they are correlated with late adult function or cognitive change. And so by doing this GWAS on longitudinal changes, what we're expecting to be able to find is only the variants that are related to cognitive aging. And that's an important point for several reasons. One, of course, being that in order to understand what mitigates risk in terms of social factors, we need to identify those risks. Another reason is that these genes can sometimes give us insights into the pathological process. And what that means is that a drug developer might say, I'm going to target that process and try to prevent cognitive aging. Well, imagine what happens if they find a gene that's associated with dementia risk, but it turns out that it's something that is only really expressed during childhood. They develop a pharmaceutical, they give it to a 70-year-old, and it has no effect. But of course it has no effect. So we want to find those genes that are actually doing the work in adulthood.
Yeah, I would say one thing though, and this is not anything criticizing, but we've dangerously looked at the ApoE relationships with education and dementia risk in the HRS. And so there is no association between ApoE and education, which is nice to know when when you're looking at education effects. So I'll end there. Someone else.
Yeah, thank you. We could talk for a long time about this stuff. Thanks so much, Mark.
So just to be mindful of time, I do see that we're at 1 o'clock. And so if folks need to drop off, that's totally fine. And Elliot, if you're free to stay to field Karen and Dana's questions, great. If you need to leave, we understand too.
Yeah, I can stick around.
I just wanted to say, my only question was to say that was such a great talk, Elliot. That was just amazing explanation of everything. And I consider it a model. I'm going to try to model my talk after you. So I just wanted to have a great thank you moment.
Oh, well, it's so good to get that sort of feedback. So thank you, Karen.
I have a question about the drug development piece that you mentioned at the beginning about the FDA's hesitancy to develop things that targeted earlier pathologies. Can you talk a little bit more about that?
Oh gosh, so the way that my understanding of the way that it works is that the FDA shows its cards a little bit in the sense that they know that there are drugs in the pipeline, not necessarily drugs, but treatments, oftentimes it's drugs in the pipeline that a company is going to want to design a clinical trial for and test whether or not they're effective. And so what they do is they kind of, in advance of receiving the data, or even those trials even commencing, they issue these papers. They're kind of like white papers in which they say, this is the sort of thing that we would need to see in order to convince us that your drug should be approved. So the companies read these white papers from the FDA, and if they were going to design a trial that, for instance, reduced amyloid levels, which is a biomarker of Alzheimer's disease, but didn't test dementia incidence rates, even if that trial went according to plan and it showed that the pharmaceutical was effective, they would know based on reading that white paper that they shouldn't do the study because they'll still won't get the drug approved, because the FDA doesn't just want to see a treatment for amyloid, they want to see a treatment that actually prevents clinical dementia incidence. So these guidelines, which are based on this cascade medical model, are they're swaying the way that drug developers are approaching the problem of Alzheimer's disease in a very strange way, because you've got groups like the National Institute of Aging Working Group saying that Alzheimer's disease begins its pathogenesis 20 or 30 years before the diagnosis is made, and that the only way that we're going to be able to intervene with Alzheimer's disease is to nip it in the bud rather than try to treat people who are on the precipice of a diagnosis. But then you've got the FDA saying that we need to see differences in diagnostic rates according to treatment and control groups, which can only really be done if you've got a drug that you can give to people who are on the precipice of the diagnosis, or if you're willing to design a clinical trial that lasts 20 to 40 years, which I don't think anyone has the appetite for, either the potential patients or the pharmaceutical companies themselves. So we're in a little bit of a big bind when it comes to promising drugs for Alzheimer's disease, because they're not going to get tested unless the company believes that they can be administered late in the disease process, which a lot of experts say isn't even worth testing because that's too late. So yeah, I mean, it's this way of thinking. This is all to say that this way of thinking about cascades and latent disease progression is having a real effect on what treatments we as patients or future patients may be getting access to.
Great, thank you so much for that. And thank you everyone for joining us. And we look forward to seeing you next week, where we will be joined by Hannah Walker from the Government Department at UT Austin. So thanks.